Hello, everyone, and thank you for joining us today. Today, we want to delve into a mini review on a significant topic in audiology, a non-syndromic autosomal dominant deafness. This is a condition encompasses a spectrum of genetic disorders leading to hearing impairment without associated syndromic features. We'll be focusing on molecular mechanisms and underlying these conditions and its pathophysiology and its epidemiologic trend.
Moreover, a molecular mechanism refers to the specific series of interactions and events at the molecular level that underlie a biological process or phenomenon. It involves the actions and behaviors of molecules such as proteins, nucleic acids, lipids, and small molecules, as well as their interactions with each other within the cell or organisms. Understanding molecular mechanism is crucial in biology, as it provides insights into how organisms function, develop, and respond to their environment. It often forms the basis for designing therapeutic interventions and treatments for various diseases.
In 85% of cases, the deafness is transmitted as an autosomal recessive trait. Autosomal dominant inheritance accounts for between 10 and 15% of cases, whereas only 1% of cases is inherited as an X-linked trait according to Orphanet. Autosomal dominant non-syndromic SNHL represents that around 20% of non-syndromic hearing loss. In contrast to autosomal recessive, dominant forms tend to be characterized by hearing loss with post-lingual onset, a progressive course, and often a milder degree of hearing loss than recessive forms, according to Shibata. Non-syndromic autosomal hearing loss accounts for approximately 70% of congenital hearing loss cases. It can be classified based on conditions patterns of inheritance, with autosomal recessive transmission being the most common, occurring in 77 to 93% of cases. Autosomal recessive non-syndromic hearing loss typically presents with prelingual onset, meaning it occurs before the development of speech and language skills. The remaining cases of non-syndromic hearing loss can be inherited in an autosomal dominant or X-linked manner, although these account for a smaller percentage of cases. The prevalence of non-syndromic hearing loss accounts varies between populations. Between 1 in 1,000 and 1 in 700 children are born with profound or severe hearing loss. Genetic factors play a significant role in non-syndromic hearing loss, with mutations in various genes contributing to its development. The identification of specific genes associated with non-syndromic hearing loss has helped in understanding its genetic basis and improving diagnosis, treatment, and support for affected individuals and families.
Molecular mechanisms that non-syndromic autosomal dominant deafness arises from mutation in specific genes crucial for auditory functions. These mutations disrupt various molecular pathways essential for hearing. For example, is the mutations in connexin genes like GJB2 and GJB6 impede intercellular communications in the cochlea, while mutations in myosin genes such as MYO7A and MYO6 that disrupt stereocilia movement in inner ear hair cells. Additionally, mutations in coherent genes like CDH23 and PCDH15 compromise the structural integrity of the stereociliary bundle. Transcription factor mutations like those affecting GATA3 can hinder proper hair cells development. Moreover, mutations in ion channel genes such as KCNO4 disturb flow vital for auditory signal transmissions.
The non-syndromic deafness causative factors are by mutations of the genes that are involved to the various functions, and their connection in deafness heavily varies. The pathophysiology of non-syndromic deafness are caused by mutation in genes that affect and causes hearing loss. A few examples like DIAPH1 mutations disrupt the actin-cytoskeleton dynamics in hair cells, affecting mechanotransduction and leading to hearing loss. Mutations in DFNA5 result in apoptotic cell death in the cochlea, leading to the degeneration of hair cells and progressive sensory neural hearing loss. Mutations in TMC1 affect the function of transmembrane channel-like protein 1, which plays a role in hair cell mechanotransduction, leading to sensory neural hearing loss.
In conclusion, non-syndromic autosomal dominant deafness represents the complex interplay of genetic, molecular, and physiological factors. By elucidating the molecular mechanisms, understanding the pathophysiology, and analyzing epidemiologic trends, we can work towards developing more effective strategies for diagnosis, treatment, and prevention of this condition. Thank you for your attention, and we'll look forward to further exploring this topic with you.